Here we have the board over here. Now let us come to the next. The Golgi apparatus or the Golgi bodies, which are also known as in plants. What do we call the Golgi bodies? Anybody has the idea? What do we call the Golgi bodies in plants? What are they known as in plants? The Golgi bodies. Golgi body or Golgi apparatus is a general name for them. Na? In plants, they are known as dictyosomes. Don't forget this name. They are known as the dictyosomes. Dictyosomes are actually those bodies in the plants. Okay, animal cell, they are known as the Golgi bodies. Okay. The specific function, these Golgi bodies are membrane-bound structures. They are membrane-bound. And membrane-bound way, they carry out many vital functions that we have many secretions present inside the cell. These secretions, they have to be packed and they have to be processed before they can be used in the metabolic processes. Maham Intasar, she wants to join the class. I don't know who Maham Intasar is, and she has left again. These are actually secretions which have to be packed. Okay? So these secretions are packed and they are processed inside the Golgi bodies, and then these secretions are released into the cytoplasm, and then they are transferred to different areas. Okay? And then they are transferred to different areas where they will be used as required. This is the main function of the Golgi body. When we look at the Golgi bodies has two faces. Okay, this is a closed tube system. It is a closed collection of tubes in which it has one face is the forming phase. It has two phases. One phase is the forming phase and the second phase is the maturing phase. Okay? There is a difference between the functions of the forming phase and the maturing phase. Let me take you to the presentation first of all, and then from the presentation we will come back to the board again. Okay? Let me take, yes. Can you see the slide better? Right. Golgi apparatus. Golgi apparatus में हमारे पास, I think I should allow Maham Intasar into the class because she is coming into the classroom. Now anybody can enter the class. Let's see the class with the Tamizi now. Okay, Golgi apparatus. Now this is somewhat what the Golgi apparatus looks like. It was discovered by Golgi in 1898. Very specific information. It was discovered by Golgi in 1898. It consists of stacks of flattened membranes, which are membrane-bound sacs, which are called cisternae. I told you the difference between cisternae and cristae in the previous period. Okay, so you very well know the difference between cisternae and cristae. Cristae, where are cristae present? Where are cristae present, beta? Can anybody tell me the answer? Where are cristae present? Give me the answer in the message box. Okay, I have an answer in the message box. The cristae are present in the mitochondria. Okay, very good, beta. It consists of stacks of flattened membrane-bound sacs, which are called cisternae. Now, these are points that you are not going to miss. It is a complex system of interconnected tubules around central stacks. Now, these are the tubules. These are the sacs, and these sacs, these are the vesicles that are going to enter them, and they are going to be packed. Uska pura process aage hum dekhenge. Cisternae stacks are continuously formed by fusion of vesicles. They are derived by the budding of the smooth endoplasmic reticulum, means materials that are produced from the smooth endoplasmic reticulum come into the vesicles, and in the vesicles they are packed and then they are stored. The outer convex surface is the forming phase, while the inner concave surface is the maturing phase. The cisternae break up into the vesicles from the latter. I'm going to explain all this on the board. Golgi complex is concerned with cell secretion. Secretions are products formed within the cell on ribosomes, then passed to the outside through the endoplasmic reticulum and the Golgi apparatus. The secretions are converted into finished product and are packed inside the membrane before they are exported. In mammals, the pancreas secretes granules containing enzymes that help in digestion. That is the cells in the mammal. The Golgi complex वहाँ पर क्या करता है? The Golgi complex has a role of formation of these granules in the mitochondria, in the pancreas. The proteins or enzymes which have to be transported out of the cell pass through the Golgi apparatus. It modifies the proteins and lipids by adding carbohydrates and converting them into glycoproteins and glycolipids. ठीक है? आगे चलते हैं. This figure, now this is a question that you should expect in your board exam. But first of all, let me go to the board and let me write down on the board what I have told you right at present. Okay? Let me go to the board and share the board with the people so that उसके ऊपर पहले मैं आप लोगों को explain कर दूँ, then उसके बाद हम लोग जो है ना वापस presentation की तरफ वापस आते हैं. Okay? I have given you the introduction of the Golgi bodies. Now let us come to the board and let me show you what happens in the Golgi bodies now. Golgi body is actually a pack of tubules like that. This is a pack of tubules. This pack of tubules, make the sketch, it is very closely connected to the endoplasmic reticulum. Okay, it is very closely connected to the endoplasmic reticulum. And the smooth endoplasmic reticulum means it is not very close to the nucleus. It is present a little bit further away from the nucleus because the endoplasmic reticulum close to the nucleus, it has ribosomes present on it. And here we do not have ribosomes. Okay? The rough endoplasmic reticulum is present away from the Golgi bodies, but here we have the smooth endoplasmic reticulum. This is the smooth endoplasmic reticulum. This is the smooth endoplasmic reticulum. And this smooth endoplasmic reticulum, the function of the smooth endoplasmic reticulum is secretion of materials. And which materials? Secretion of fats, secretion of liquids, production of proteins, and all these things. Now, from the smooth endoplasmic reticulum, materials are going to be produced. This is one phase of the Golgi apparatus, and this is the second phase of the Golgi apparatus. This phase of the Golgi apparatus is facing the smooth endoplasmic reticulum. This phase of the Golgi apparatus is facing the away from the smooth endoplasmic reticulum. Now, what happens? Okay, vesicles or little substances are produced from the smooth endoplasmic reticulum. Okay. Little droplets and little vesicles are produced from the smooth endoplasmic reticulum, but these droplets are not processed. They have to be processed and they have to be stored. So what happens here, that these droplets, ये protein है, lipids हैं, anything, ठीक है? ये जो droplets हैं, they are produced on this side from the smooth endoplasmic reticulum. They are going to come into the forming phase. This is the forming phase of the Golgi apparatus. This is the forming phase of the Golgi apparatus. This forming phase is going to receive these vesicles and these droplets from the smooth endoplasmic reticulum. It is going to pack these droplets and then it is going to release them from there after processing them and after packing them in special materials. Okay, in special membrane, it is going to release them after packing them. कैसे पैक किया इनको? आप यूं समझ लीजिए, थोड़ा सा कोशिश करती हूँ कि ये इस तरह इनकी पैकिंग भी हो गई. They have been processed and sorry बेटा, थोड़ा सा pen is not coming in control. ये ठीक है, ये थोड़ी सी पैकिंग होगी and this is the maturing phase. This is the forming phase. This is the maturing phase. Vesicles and droplets produced from the smooth endoplasmic reticulum come and attached at the forming phase inside the Golgi apparatus, which is also known as the dictyosome. These are going to be processed inside the Golgi apparatus, these vesicles and droplets, and they will be packed inside a specific membrane, and then they will be passed out. Now, these are either going to be circulated into the cell, into the cytoplasm, or they will be sent out of the body. जैसे कि हमने pancreatic enzymes का भी पढ़ा है कि the enzymes they are produced and they are sent out. Now there is a very important question that you should expect in your board exam, because this time ये question जो है ये board exam में नहीं आया था. तो वो जो क्वेश्चन है वो क्या है वो हम देख लेते हैं. ठीक है, मैं आपको क्वेश्चन लिख देती हूँ. लेट मी सी हु वी हैव इन द क्लास. ओके, ठीक है. आई एम enabling the classroom again so that anybody that wants to enter the class should seek my permission. First of all, Mariam Intasar, you have joined the class. Welcome, beta. Are you a new admission, beta? Message box में जवाब दे दीजिए मुझे. Okay. यहाँ पर अब हम देखते हैं. The question that you should expect is draw, and this is an 8-mark question. Draw a clear. Write down the question, beta. Draw a clear. What is happening? Draw a clear diagram to show the relationship. Write down this question. To show the relationship between the endoplasmic reticulum, Golgi complex, and cell cytoplasm and cell membrane. Draw a clear diagram to show the relationship between the endoplasmic reticulum, the Golgi complex, and the cell cytoplasm and cell membrane. ठीक है, ये जो क्वेश्चन है बेटा ये एक लॉन्ग क्वेश्चन में आता है. इट कम्स विद अ लॉन्ग क्वेश्चन. ठीक है, नाउ लेट अस गो बैक टू द नेक्स्ट स्लाइड. नाउ आई विल शो यू दिस स्लाइड. इसका स्क्रीनशॉट आप लोगों ने ले लेना. यू हैव टू टेक द स्क्रीनशॉट. ठीक है, दिस फिगर शोज द रिलेशनशिप between the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the plasma membrane. This is a very, very important sketch. Okay. The Golgi apparatus has two ends, forming phase and maturing phase. Okay. Blebs from the tips of the smooth endoplasmic. Blebs का मतलब होता है pieces, cutouts. From the tips of the smooth endoplasmic reticulum fused with the Golgi apparatus cisternae at the forming phase, whereas secretory granules, which are known as transport vesicles, are pinched off from the maturation phase of the Golgi apparatus. Now look here. This is the Golgi apparatus. Now this is the Golgi apparatus. ये हमारे पास क्या चीज है? This is the Golgi apparatus. Golgi apparatus में एक हमारे पास होता है cis phase and एक हमारे पास होता है trans phase. The cis phase is another name for the forming phase. Write it down. The cis phase is also known as the forming phase, and then the trans phase is also known as the maturing phase. Now this is the smooth endoplasmic reticulum, and from the smooth endoplasmic reticulum these vesicles are pinched off. These vesicles are secreted inside the smooth endoplasmic reticulum, and they are pinched off from the smooth endoplasmic reticulum, and these vesicles are transported to the Golgi apparatus. ठीक है, in the Golgi apparatus now they are going to be processed, and after processing they will be packed, and now they will be converted into the secretory vesicles. ठीक है, the secretory vesicles over here they are the lysosomes, which can be enzymatic in nature. ठीक है, इसमें आपके पास you have enzymes की collection. इसके अंदर आपके पास जो secretory vesicles हैं, which are generally known as the lysosome. These are packed and they are released from the trans phase, which is also known as the maturation phase. And then from there it comes to the cell membrane, and it is either released out of the cell into the space outside and transported to various areas, or it is circulated inside the cytoplasm if the requirement is there in the cytoplasm. If you have any questions, Rita, You people should ask me. Let us go back to the question that I wrote on the board for you people, and then we will come back to this diagram again. Question hai, draw a clear diagram to show the relationship between the endoplasmic reticulum, the Golgi complex, and the cell cytoplasm and cell membrane. यहाँ पर we can add cell cytoplasm, cell membrane के साथ we can add lysosomes also. This is a very important long question, and you people must expect this long question. Okay, this long question did not come this time in the paper. You should expect this long question. इसका आंसर देख लीजिए. 1 minute, beta. Just give me 1 minute. 1 minute. This figure shows the relationship of endoplasmic reticulum with Golgi apparatus, lysosome, and plasma membrane. Golgi apparatus has two ends: the forming phase and the maturation phase, the cis phase and the trans. Blebs from the tips of the smooth endoplasmic reticulum fuse with the Golgi apparatus cisternae at the forming phase, whereas secretory granules are pinched off at the maturation phase of the Golgi bodies, and then they are transported to different parts of the body. You are going to give a detailed explanation of this. However, this diagram is very much relevant to the question that I have given you. Okay, now let us go back. The next lesson is lysosomes. We will not discuss lysosomes today. Let us revise the Golgi apparatus. Golgi apparatus, it was discovered by Golgi in 1898. It consists of stacks of flattened membrane-bound sacs called cisternae. It is a complex system of interconnected tubules surrounded by surrounding central stacks. Cisternae stacks are continuously formed by fusion of vesicles derived by the budding of the smooth endoplasmic reticulum. Their outer concave convex surface is the forming phase, while the inner concave surface is the maturation phase. The cisternae break up into vesicles from the latter. Golgi complex is concerned with cell secretions. Secretions are products formed within the cell on the ribosomes, then pass to the outside through the endoplasmic reticulum and the Golgi apparatus. The secretions are converted into finished product and are packed inside the membrane before exporting out. Some examples in mammals. The pancreas secretes granules containing enzyme that helps in digestion. The Golgi complex has a role in the formation of these granules. The proteins or enzymes which have to be transported out of the cell pass through the Golgi apparatus. It modifies the proteins and lipids by adding carbohydrates and converting them into glycoproteins or glycolipids. This is a very important diagram. इस diagram को आप देख लीजिए. If you have any question, आप मुझे message box में question दे सकते हैं. Any question at all, you can ask me in the message box और मैं आप लोगों को फिर क्या करूँगी, आप लोगों को जवाब दूँगी. Any question.